Hello, everyone, and welcome to the BioProcess International Fall Digital Week, brought to you by the producers of the online and face-to-face BPI events and content series. My name is Brianna Cosafret, and I'll be your host for today's session titled Unlocking the Future of Biologics Manufacturing, Cas-CLOVER for Big Pharma and Advanced Pipelines. First, I'll cover some quick housekeeping items. If you experience difficulties with audio or advancing slides, refresh your screen with F5. If you are experiencing other issues, hit the question mark button to receive assistance. At any time during the presentation, please submit your questions into the Q&A window on the left-hand side of your screen. And lastly, in 24 hours, you'll receive a link to watch a recording of this session. Let's now begin by introducing our speakers for today. We have Jack Crawford, CEO, and Corey Brizzee, Ph.D., the Director of Gene Editing, both from Demeetra. Welcome, Jack and Corey. I'll now hand it over to Jack to begin the presentation.
Thank you. Thank you for everybody who's attending this presentation. I am going to go over the unique freedom to operate advantages of Cas-CLOVER, as well as the business advantages, while my colleague Corey Brizzee will go over the details on technology and applications. First, a little bit about Demeetra. We're based in Lexington, Kentucky. 80% of our people are hands-on research. And this matters because it allows us to continuously innovate in gene editing. And we also are able to transfer validated know-how to our clients so that they can take these technologies and use them right off the bat effectively and quickly. Another thing that we've noticed that holds back innovation and development production is licensing, commercial licensing. So we've also developed some very streamlined commercial licensing options and methods, which we'll talk a little bit about later. So first, an overview of the core technology we'll be talking about and why we are able to offer, I think, uniquely comprehensive freedom to operate for cell line development in precision gene editing. This is Cas-CLOVER. It works with two guide RNAs. The guide RNAs complex with dCas9. This is a completely nuclease inactivated dCas protein, and it binds to the target site, and it only binds, it does not cut. And it's fused to a dimeric nuclease called CLO51. CLO51 will only cut the genome when both the left and the right guide RNAs come together and bind at the target site. This is what makes Cas-CLOVER more precise than CRISPR-Cas9. So it's easy to use compared to other technologies like zinc finger nuclease and TALENs, which require extensive protein engineering because you use guide RNAs, which are simple and easy to produce, and there's many providers of those guide RNAs. This technology provides higher efficiency and, like I said before, much better precision compared to CRISPR-Cas9, and it also produces more complete knockouts with higher targeted integration compared to CRISPR-Cas9. My colleague Corey will talk about that in detail. Now on to our freedom to operate. So every single component of this technology is covered by our worldwide patents. So the first components, the guide RNA and the dCas9, we have an in-license from ERS Genomics, which licenses the foundational patents from the California, Vienna, and Charpentier group, the CVC. This is important because this group has the most solid and the best IP on fully nuclease inactivated Cas9 proteins. That's extremely important for us because the only Cas protein that we use is a dCas9 protein. We also use what's called the two-part guide RNA system. Second, the CLOVE51 is a dimeric nuclease, which was invented and patented by the Helmholtz in Munich in Germany. This endonuclease is exclusively licensed by Demeetra for cell line development and bioproduction. In addition, the entire system is patented by 𝐭𝒂𝒏𝒈 Therapeutics, whom we also have an exclusive license from. And as I said, Demeetra is continuously innovating our gene editing technologies and our suite of specifically edited 𝐤𝒑K1 cells for bioproduction. As part of that, we're constantly improving our technology, and we've also improved the enzyme CLOVE51 and patented that. What's unique about what we're able to offer is all of these different buckets of intellectual property and patents put together in one simple, streamlined commercial license. And one thing I want to distinguish before I move on to the next slide is that, remember, with Cas-CLOVER, you're editing the genome only with CLOVE51, and we're only binding to the target site with dCas9. That's extremely important because Cas9 is an extremely crowded and conflicted intellectual property landscape. For example, when you search in a patent search engine, the one I use is called the lens.org. When you search for title, abstract, and claims for Cas9, you retrieve close to 20,000 patent records and close to 12,000 simple families of patents. The foundational patents don't expire until at least 2033, and other popular Cas enzymes also have this same crowded issue. There are at least 500 individual applicants of Cas9 patents. This means that even with multiple licenses, freedom to operate remains very uncertain with CRISPR-Cas9 for commercial use. Also, initial and ongoing due diligence imposes a very, very significant resource burden on companies trying to use this technology for commercial purposes. On the other hand, CLOVE51 is clean and clear, a very stark difference. Remember, we edit the genome with CLOVE51. When you do that same exact search in the lens.org, you get 36 patents, 20 families. And the best part about this is there are 3 total individual applicants. One is us, Demeetra, the other Helmholtz Munich, and the other 𝐭𝒂𝒏𝒈 Therapeutics, all of which we have exclusive licenses for cell line development and bioproduction. This provides a simplified option for diligence, solid intellectual property, and delivers clear freedom to operate. The next point that I'd like to make is how we offer our clients the ability to unlock cost certainty, especially for large biologic pipelines. In the Demeetra licensing model, it's a one-time pipeline-wide commercialization access fee. Our competitors charge large yearly fees and milestones, and oftentimes they also impose royalties on the end product. So a simple demonstration of what this looks like is as the pipeline gets bigger from 5, 15, and 30 programs, the cost is extremely higher. It increases with the amount of innovation that your company does, whereas the Demeetra cost and licensing model is relatively flat across all these pipelines. Therefore, our model scales with innovation, not against it. This next section and point that I'd like to talk about is what we call every clone counts. So we've heard this in the industry that people want to eliminate the costly handoff between R&D and production. So The problem with Cas9 and legacy systems is that you've got this uncertain freedom to operate situation for commercial use. Therefore, some companies are using Cas9 or other Cas technologies to do their discovery or possibly their development, and switching to another technology or something completely different for the manufacturing phase. That requires re-engineering of the cell line or licensing. It leads to delays, variability, and loss of validated clones. Each handoff compounds the time and the cost risks. So at the end of the day, switching technologies always adds time and costs. You lose value and you increase the risk of failure. The solution we provide is Cas-CLOVER, which can be used as a single platform for both discovery and manufacturing phases. You use the same editing technology, therefore you get the same editing efficiency and the same IP coverage from bench to bioreactor. There's no need to rebuild or try to license new cell lines or switch to a less effective technology. And importantly, it preserves every validated clone and the investment behind that. We've heard people talk about this in depth, where you have a really great clone in discovery, you try to remake that for manufacturing, and the clone just doesn't produce the same. With our system, you have seamless integration across all phases, no rebuilds, no lost value, and no added risk. We're now introducing 2 upgraded platform purchase options. As I said before, we're constantly innovating this technology and adding new technologies. And so we are building out not only our standard perpetual license, which is unlimited forever commercial use for the current technology. This is a one-time fee. We give technical support for the first 12 months. We're now offering what we call the Enterprise Platform Subscription 36. It's a 3-year subscription payment that converts automatically to a perpetual license. What this means is that you can have the option to have a smaller 3-year fee. So you spread the cost out over 3 years, and it ends up being 20% more than the one-time fee, but you get that added spread of the cost. The other advantages of using this option is during that 3 years, we're going to be innovating and making new types of technologies. You automatically get access to the latest of our technologies throughout that term. The other benefit is we give a reagent credit of 60 vials of either Cas-CLOVER or Harbor-in transposase, which my colleague will talk about in detail, for stable cell line production. If you choose this option, we provide technical support through the whole term. And like I said, it automatically converts to perpetual license, and you can always elect early pay to convert. With that, I'm going to turn it over to my colleague, Corey, who will tell you more about the technology and the applications.
Thank you, Jack. I think that the commercial access to these gene editing technologies is really going to be important when we start talking about the top trends that are going into 2026. So a lot of the bioprocessing conferences that we have attended, there's a lot of talks about large data omics, whether that's going through genomics, transcriptomics, or proteomics. How do we utilize AI and machine learning to really parse through this large data set to identify inhibitory enzymes faster and how to circumvent those problems in large-scale processes? I think all roads lead to genetic engineering. This could be through creating knockouts and prototrophies. This helps CHO cells overcome amino acid limitations during your fed batch in your large-scale processes. This also allows you to have a selection mechanism for your CHO cells to increase antibody titers. You can also see a lot of trends that are leaning towards genetic engineering that reduces inhibitory byproducts from amino acid metabolism and also improving strategies to mitigate nutrient balance and product quality. And this is what I call a pathway flux mechanism. So a lot of people are very known to familiar with glutamine synthetase knockout, which creates a selection mechanism for CHO cells. But you can also create knockouts in pathways. So limiting the branched-chained amino acids that might pathway flux into more production that eliminates lactate buildup for increased titer production for your antibodies. A lot of Demeetra cell line platforms allow for this to happen. So our Cas-CLOVER targeted nuclease, as Jack previously mentioned, is high efficiency, high precision compared to CRISPR-Cas9. It creates more complete knockouts, and it also has a higher targeted integration efficiency. Our clean-cut GS CHO cell lines are built off of the Cas-CLOVER targeted nuclease. This enhances product quality and yield, and it also enables new modalities to be made, ADCs, especially antibody engineering, such as afucosylation and different glycosylation patterns. We also have our Harbor-N transposase, which is based off of the piggyback transposase, and this is very well-known transposase that integrates stably at highly expressed sites for high titers, and is very well known for being able to support large cargo capacities. And this really becomes important when we try to build more complex molecules beyond a heavy chain and a light chain. So fortunately for Demeetra, we are able to produce recombinant protein that allows us to reveal the cutting mechanism of Cas-CLOVER compared to CRISPR-Cas9. So as Jack previously mentioned, we utilize an inactivated Cas molecule fused to our proprietary CLO51 nuclease domain. And the CLO51 nuclease domain is an obligate homodimer. So it actually needs its partner to actively cut the genome. This is different than the monomeric CRISPR-Cas9. So we can incubate our recombinant protein with genomic DNA that has our target site in mind. You can incubate that as well with the left guide that has your PAM site and your right guide with your PAM site. And then you can look at the cutting products from either Cas-CLOVER or Cas9. When you sequence these, you can see that Cas-CLOVER cuts in a staggered cutting mechanism in between the left guide and the right guide. This is very different than the typical Cas9 cutting mechanism that is very typically 3 base pairs from the PAM site, and it's a blunt cutting mechanism. We are seeing improved knockout efficiencies in CHO cells. This is a very popular target, glutamine synthetase, which will be mentioned several times in the next couple of slides. With Cas-CLOVER, we are able to utilize Cas-CLOVER with a variety of guide RNAs. This could be the 2-part guide RNA, which has the crimp RNA and the tracer RNA, which you would pre-complex prior to transfection. And then you also have the single guide RNAs that are covalently attached from either IDT or Synthego. And these are very consistently producing 75% editing efficiency of this pool with whatever different guide pairs that you would like. This is compared to Cas9, which is around 55%. We are also seeing improved knock-in efficiencies with Cas-CLOVER, and we attribute this to the staggered cutting mechanism that Cas-CLOVER produces. So this is in HEC293 cells, we're able to have a GFP plus a puromycin selection. This knock-in cassette is around 7 kb. With Cas-CLOVER, without any selection, and this is just post-transfection, around 10% of the pool has a knock-in, where this is compared to Cas9, which is only around 3%. You are able to enhance or enrich this cell pool up to 70% compared to Cas9, which is only around 30%. Now, I mentioned briefly that there's a lot of off-targets associated with CRISPR-Cas9, and our sister company, Poseida Therapeutics, ran a off-target analysis. This is with 𝐦𝒑 compared to other gene editing technologies. And as you can see, compared to other gene editing technologies, Cas-CLOVER was by far the lowest gene editing technology that produced the lowest off targets. CRISPR-Cas9, as having a blunt cutting mechanism, often also produces a high translocation rate. Utilizing 3 different approaches to identify translocations, they were able to see over 6% translocation events with any different method that they looked at for translocation rates with CRISPR-Cas9. Whereas with Cas-CLOVER, in T cells, there was no translocation events with Cas-CLOVER. So now that we're utilizing a really good gene editing technology, we're able to produce a nice commercial clean-cut GS cell line platform for bioprocessing. So we were able to adapt 𝐭𝐤1 cells to suspension and again attack glutamine synthetase for a knockout to provide selection. The main gene of glutamine synthetase resides in chromosome 5. However, there's been a recently identified pseudogene of glutamine synthetase in chromosome 1. We can utilize the specificity of Cas-CLOVER to target each of these genes separately and sequentially to provide a more stringent selection system. Here, the blue guides for the main gene of chromosome 5 will not work because there's an intron in the middle of these guides. So the distance is too far for Cas-CLOVER to cut, whereas the red pair of guides will cut the main gene at chromosome 5. And vice versa, on the pseudogene, we can utilize that disappearance of the intron where the blue guides will now be in vicinity of each other and you will see effective cutting, where the red guides that would work on chromosome 5 are not going to work on the pseudogene on chromosome 1 because there's several mismatches. So we tested this theory and we utilized the blue guides targeting chromosome 1. The wild type band or non-edited band of the pool resides here, and you can see a nice percentage of edited fraction from this pool, whereas this same guide pair did not target chromosome 5 and did not cut at this target because of the spatial difference. This also points to utilizing the specificity of Cas-CLOVER with genes that are very homologous to one another. Cas-CLOVER also produces large deletions compared to CRISPR-Cas9. So some of our clones that were produced from these pools actually showed deletions of 16 base pairs, 26 base pairs, and 28 base pair deletions, and they're very easily characterizable throughout your process. With these bigger deletions, we're seeing a more disruptive knockout. So we grew these clones with glutamine, and they are growing just like the 𝐭𝐤1 suspension cells. Without glutamine, these clones are dying within 9 days, and this is complete cell death. So there's no residual viability with both of these glutamine synthetase genes knocked out. and this manuscript was submitted to the Biotechnology Progress Journal in the last month. So now we're going beyond that parental cell line for 𝐭𝐤-𝐂𝐚𝐬-GS, and now we're producing derivative cell lines that could have some niche applications for a variety of different products. So we went beyond a glutamine synthetase knockout and produced glutamine synthetase plus a FUT8 knockout, and this is what we're terming 𝐭𝐤-𝐂𝐚𝐬-GS ADCC+. So this will create an afucosylated antibody that enhances your antibody-dependent cellular cytotoxicity. So targeting the FUT8 gene with Cas-CLOVER, we were able to identify a very good guide pair that cuts around 80% of the pool. This is compared to around 55% with Cas9. So having a pool that's already 80% edited for the FUT8 gene, we were able to do LCA selection, and that is utilizing 𝐸𝑛𝑠-cholinesterase agglutinin, which is a lectin. This lectin binds to fucosylated surface proteins on the cell, and during selection, that binding of the lectin to the fucosylated surface proteins will actually kill the cells. So then in post-selection, our pool was 98.5% FUT8 positive knockouts. And we were able to actually sort that from staining with LCA, and now we can isolate a 100% population that had FUT8 knocked out. And after single cell cloning, we were able to identify a lot of different clones for the FUT8 knockout, and we were able to provide a frequency distribution of these clones for the indels that they have. You can see a lot of these indels for Cas-CLOVER. This is very indicative of Cas-CLOVER editing, where you have large deletions throughout that FUT8 gene, where compared to CRISPR-Cas9, you would probably see a large population between 12 base pairs to 1 base pair deletions with CRISPR-Cas9. So we have a couple of clones that have full exons deleted, and this is owing to the more disruptive fucosylation knockout. We are also creating a clean-cut-GS delta host cell proteins, or HCPs. Host cell proteins can affect the production of antibodies in CHO cells. They can also affect the purification and the efficacy of the antibodies you're trying to purify from CHO cells. This could be properties accounted by proteases, lipases, deamidases, host cell proteins that are affecting the redox properties, and this can lead to your drug being degraded or neutralized. So we actually targeted 3 host cell proteins, A, B, and C. We utilize a couple different pairs to target each host cell protein. We identified the top pairs for each host cell protein, and then we utilized the multiplex for 3 host cell proteins and 1 transfection. So we utilized the Lonza nucleofector, and now we have a pool of cells that are edited in different ways, different combinations. There's no good selection for host cell proteins, so we went right into single cell cloning, and then we were able to perform a multiplex PCR that allowed us to identify clones that had different variations of host cell proteins knocked out. You can see the top 2 rows are a single knockout at host cell protein A. You have different combinations of 2 host cell proteins knocked out in the pink and the orange. And then you also have a potential for a triple knockout of all 3 host cell proteins. Now that we have really good genotypes of these cells, we want to look at how these edits are relating to the protein expression. So we're able to take our clean cut GS cells as our control. You can see the host cell protein A or B is identified here. And then we look at the clones that have the biggest disruption of protein expression. So this allowed us to select those clones that had the biggest disruption or abolishment of that protein of interest. We were able to utilize the HARBOR-N transposase to stably integrate either trastuzumab or a more complex molecule, a masked trastuzumab, stably into these clones. And then we were able to perform a 24 deep well plate fed batch experiment, isolate the antibodies from the supernatant, and then look at the production of the heavy chain and the light chains for these. Our clones were able to produce trastuzumab just as equally as our clean cut GS cell line. And it was also able to make a more complex molecule, such as this mass IgG at 2 grams per liter, which has the major species as the heavy chain and mass light chain. So there's no cleavage of that masked nanobody. And then you can also see a slight bit of cleavage of the mask from the light chain. And the good thing is that this was a single host cell protein knocked out, and we're looking to have the further host cell proteins that were knocked out identified and analyzed with LC-MS. Utilizing Cas-CLOVER, we're able to provide a nice clean-cut CHO platform pipeline. Now you can see there's a variety of CHO cells listed here. I mentioned the clean-cut GS, the clean-cut GS ADCC+. We're also developing a clean-cut dual select. So this is adding another selection system. This is the DHFR for higher productivity. This is going to be very key for complex molecules and bispecifics. We're also planning on launching an ADC-ready cell line. This is a dual knockout utilizing the MGAT1 knockout gene for antibody-drug conjugates. And realistically, with Cas-CLOVER, you can utilize this beyond just CHO-K1. You can utilize Cas-CLOVER for your CHO host or whatever host system that you use to create a pipeline like this as well. So HARBOR-N is our another core technology from Demeetra. This is very critical for cell line development. This is a license-free commercial transposase. The transposase is an mRNA format, and this is an optimized mRNA format, which prevents degradation and facilitates higher copy numbers. The transposase will recognize the inverted terminal repeats from the transposon. It will excise the cargo and then integrate it into the genome at TTAA sites. And this is very well known for being very stable integrations. With a first-time offer, we're offering the mRNA with 5 cell line development tailored transposons. These are banked at GenScript for stress-free cloning. So we have a lot of transposons that have the core components, which is the inverted terminal repeats, as well as the core insulators. You also have multi-cloning sites for popping your gene of interest into these transposons. You have another transposon that has glutamine selection. So if you do have a CHO host with glutamine synthetase knocked out, you can utilize this for cloning your heavy chain and light chain, or whatever chain or molecule it is in there. You also have some plug-and-play transposons, such as D04 and D08. These are able to be utilized right away with HARBOR-N. This D04 has GFP with puromycin selection. So you're able to use that and then do some puromycin selection and see GFP fluorescence and GFP positive cells. You're also able to use 𝐷08, which has trastuzumab, with our glutamine selection cassette. This allows you to rapidly go into a CHO host with glutamine selection and produce trastuzumab stably. And our other offer is transposon is 𝐷09. This is a cloning cassette that's very similar to 𝐷08, but it allows you to clone in your heavy chain of interest and your light chain of interest to produce your antibody of interest. So our GS platform was made with Cas-CLOVER. So the Cas-CLOVER generated the glutamine synthetase knockouts in suspension CHOK1. We're able to stably express trastuzumab using the license-free HARBOR-N transposase. And this really reveals stable, high-titer cell lines, whether it's the pool or the clones. So over 60 generations, we were able to track our antibody titers. From Gen 0 to Gen 60, you see really no fall off. So really consistent stability from 60 generations of the pool for antibody titer, as well as cell-specific productivity, which utilizing HARBOR-N transposase, we are consistently seeing over 40 picograms per cell per day in cell-specific productivity, which is industry leading. This transfers quite well to the clones because these clones reflect of the pool's nature. So our clones that we identified have very good stability over 60 generations, and their cell-specific productivity remains in industry standards. Now, I will mention that Demeetra only has a 24 deep well fed batch capability and small scale capability for fed batches, and our viable cell densities from these were only around 10 to 13 million cells per mil. When we're able to utilize collaborators and partners, these bulk pools are able to get over 8 grams per liter with trastuzumab in bioreactors or small systems like the Amber 250. We're also able to identify very stable copy numbers over 60 generations. So we took this pool and utilized dPCR and identified around 14 copy numbers on average throughout from Gen 0 to Gen 60. So these integrations that the HARBOR-N transposase is making are very stable throughout time. This really will reduce your pipeline timeline with stable bulk pools. HARBOR-N can be used to produce material for IND-enabling tox and phase 1 studies, and you can actually, while you're making this material, now you're able to go in and do things that create a little bit longer timelines for bioprocessing. This could be clone generation and expansion, characterization and banking, and while you're making material, you're still able to do stability and titer studies, saving a company lots and lots of time and uncertainty throughout their cell line development processes. I would like to thank you for giving us the time to talk, and I will open the session up for questions.
Great. Thank you very much, Corey and Jack. All right, here's our first question. What exactly is covered by the Comprehensive Freedom to Operate Cell Line Development Package with respect to Cas-CLOVER? Does it include CVC guide RNA and dCas9 claims and the CLOV51 related improvements? Do you have legal opinions?
Thank you. I'll take that. This is Jack. Yes, so the license that we provide combines all of those components. So yes, guide RNA claims, dCas9 claims, CLOV51. If you get the EPS36, you also, you also get the improvements we've made to CLOV51, and then the EPS36 subscription will also provide future improvements as well. And yes, we do have legal opinions. We've done a lot of research on this, and our opinions come from global, very, very reputable law firm.
Great. Thank you, Jack. Our next question here. Do you plan to expand your CHO host cell protein knockout program to include proteins that interfere with downstream purification?
Yeah, I can take that. This is Corey here. We're currently exploring a multitude of host cell protein knockout cell lines. This is creating panels of different versions of host cell proteins that can affect different processes and different steps during the process of production and purification. So we're attacking things like lipases and some other sticky host cell proteins throughout the process.
Yeah, it's a sticky situation that we're trying to get out. But just to add to that, yeah, so we've heard from our clients that this is a big problem, and if they encounter a failure due to host cell protein purification issues, it costs possibly tens of millions of dollars. So we're really focusing on this as our next area of development, and there is literature on it, but we don't know if we knock out, like, let's say 20 host cell proteins, is that going to affect the cell in its ability to produce your protein of interest? So we're going at it in two different ways: the suite of single or maybe double knockouts or maybe triple knockouts, and then possibly one that's got 10 or more knockouts.
Right, very good. Thank you both, Jack and Corey. We have time for one more question here. Can you share the origin of your 𝐤𝐭1 knockout line and confirm whether a complete history and viral testing documentation are available?
Yeah, I can take that one. The 𝐤𝐭1 that we have engineered is from the 𝐂𝐚𝐬, the European Collection of Authenticated Cell Cultures. So it's one of the globally recognized authenticated 𝐤𝐭1s. And we have a great commercial license from them. And actually, we just expanded that commercial license to make sub-licensing of these edits in this 𝐤𝐭1 very streamlined, something that our clients have huge headaches with other CHO hosts. So that's the origin. We've got the entire history reports from vial crack to freeze down, everything that's touched this cell. We've got the COAs. We've done mycoplasm sterility testing, and I'm pleased to say that we just did 2 different panels of viral clearance testing, and they all came back clean. So we're very happy with this cell line.
And that's all the time we have for questions today. Thank you very much, Corey and Jack, for a great session. If anyone submitted a question that wasn't addressed, keep in mind that the speakers will reach out to you directly. This session was recorded. You'll receive a notification in 24 hours when the on-demand session is available for viewing. And before you log off, please take a moment to complete the feedback form so we can continue to improve your Digital Week experience. On behalf of Informa Connect Life Sciences, thank you and have a great day.